Today we will be teaching you about mitosis and meiosis, their stages, how they function, and their importance in cellular life.
During interphase, the genetic material forms a tangled mass called chromatin. The nucleus is bounded by the double nuclear membrane and contains nucleoli. This is not a stage of mitosis. The cell spends the majority of its lifetime in this stage.
During prophase, the chromatin starts to condense into chromosomes, the nucleoli disappear, and the nuclear membrane begins to break down. The centrioles migrate to opposite poles, and the chromosomes move towards the center of the cell. The meiotic spindle starts to appear.
The spindle forms, and the chromosomes take up position in the middle of the cell. Each double-stranded chromosome is attached to a spindle microtubule at its centromeres.
Anaphase begins when the centromeres of each chromosome separate. Each sister chromatid now has its own centromere and is called a chromosome, so there are twice as many chromosomes as there were during metaphase.
In telophase, the chromosomes gather at the poles and begin to form daughter nuclei. The two daughter nuclei will be identical to the nucleus of the parent cell. The nuclear membrane begins to reform, the nucleoli begin to reappear, and the chromosomes begin to unwind into chromatin. Cytokinesis accompanies telophase. In animal cells, the membrane constricts in a ring around the middle of the cell and pinches the cell in two. This ring is called a cleavage furrow. The cytoplasm and all of its constituents are passively divided so that each cell gets about half the materials and organelles in the cell.
At the end of mitosis, two daughter cells identical to the parent cell are produced. Mitosis is important as it allows the cell to grow and repair in multicellular organisms. In binary fission, this is the method of reproduction in single-celled organisms.
The purpose of meiosis I is to separate homologous chromosomes. During prophase I, the chromatin starts to condense into discrete chromosomes, the nucleoli disappear, and the nuclear membrane breaks down. The duplicated homologous chromosomes form tetrads, and crossing over, an exchange of genetic material, occurs at locations called chiasmata.
During metaphase I, the tetrads line up along the center of the cell and the microtubules attach to the kinetochores. Independent assortment in the random alignment of the tetrads occurs. This causes the gene alleles to separate independently from each other.
As the mitotic spindle shortens, the homologous chromosomes separate and are pulled to the cell's poles. The sister chromatids, however, remain attached, and the cell still has the same number of chromosomes.
During telophase I, the homologous chromosomes are at opposite ends of the cells. The nuclear envelope reforms around each chromosome set, the spindle fibers disappear, and cytokinesis follows with the formation of a cleavage furrow, resulting in the pinching of the cell into two cells. After cytokinesis, each of the two daughter cells has a nucleus with a haploid set of replicated chromosomes.
The purpose of meiosis II is to separate sister chromatids. During prophase II, the centrioles again migrate to the poles, the mitotic spindle begins to form, and the chromosomes begin to move to the center of the cell.
During metaphase II, single chromosomes align on the center of the cell again as in mitosis, and the spindle fibers attach to kinetochores. This is in contrast to metaphase I, in which homologous pairs of chromosomes align in the center of the cell.
In anaphase II, the centromeres separate and the two chromatids of each chromosome move to opposite poles in the spindle. The separated chromatids are now called chromosomes.
During telophase II, the nuclear envelope reforms around each set of chromosomes. The spindle begins to disappear and the chromosomes start to unwind. Cytokinesis takes place, producing four daughter cells, each with a haploid set of chromosomes. Because of crossing over and independent assortment, the purpose of meiosis is a split of the amount of genetic material in half so that it can be combined for sexual reproduction, a process crucial to the variety of life on this planet.
Both meiosis and mitosis are found in complex organisms which reproduce sexually. Mitosis may be used for growth, the replenishment of depleted organs and tissues, healing, and sustenance of the body. Identical versions of cells can be created to form tissues through mitosis. Meiosis is a special process reserved for the creation of egg and sperm cells, otherwise known as gametes. The same patterns may be found in many species of plant and animal cell reproduction.